So we have been talking about DNA for a while, right? And so we know that DNA is one of our macromolecules. We know that DNA is the molecule that carries our genetic information. What we mean by that is that it carries the instruction for passing on traits, passing on genes, and it passes these instructions to new cells. It can do this through mitosis or through meiosis. What we really haven't talked about, though, is how is it that these instructions get carried out? Because again, you can give all the instructions in the world, but somebody has to carry out the instructions. In other words, how do genes actually work? All right, so the DNA molecule has 4 nitrogen bases. We know this, right? Adenine and thymine, they're base pairs. Cytosine and guanine, also base pairs. The order of these nitrogen bases is the code for all proteins that are made in the cell. So if I have AAA, CCC, GGG, that might be the code to make brown eyes. That might be the code for the protein that is going to give you freckles. Who knows, right? Genes are made up of DNA. Remember, genes is just a section of your DNA that codes for a particular trait. A gene is coded DNA instructions, and it controls the production of specific proteins. Remember, ladies and gentlemen, way back when we did our macromolecules, we said that there were carbohydrates, those are used for energy. We said that there were lipids, that those were used to store energy and to make waterproof barriers. We said that there were nucleic acids, and those were to store our genetic code. And then we said, and then there's proteins, and they do everything else. DNA basically is the code to making proteins. Why? Proteins make enzymes. Proteins are structural. Proteins carry oxygen throughout the body. Without proteins, life really doesn't exist. So it is those proteins that the DNA is coding for us to have our traits and to be able to carry out all of our life functions. So when we say a gene carries a trait and we can see it, we call that gene expression. We talked about this when we talked about Mendel a while ago. So this purple flower is expressing that gene. It might have what's it called? A gene inside of its DNA for whiteness, for be a white flower, or maybe to be a blue flower, or maybe to be a red flower, but the one being expressed is purple. In other words, the DNA is telling somebody to make the protein that makes it purple. In order for a gene to actually be expressed, 2 things have to happen. Number 1, there has to be transcription of the DNA. And number 2, there has to be translation of the DNA. Now, in order for there to be transcription and translation, in order for a gene to be expressed, that's where RNA comes in. Translation and transcription does not happen without RNA. So, 3 critical differences between DNA and RNA. Let me just start by saying this. They are both nucleic acids. They both have adenine, thymine, and cytosine. They have that in common, okay? But what are some differences? Critical difference number 1. RNA is single-stranded. DNA is double-stranded. That is critical difference number 1. Critical difference number 2. RNA contains the sugar ribose. DNA contains the sugar deoxyribose. These are slightly different in terms of their structures, and you can see those structures here on this picture. Okay, so we can see in the picture, double strand for DNA, single strand for RNA. We see the double base pairing on DNA. We see a single base pair, or a single base, I should say, on the RNA, and then we see the different sugars. And then the final and probably most critical difference between DNA and RNA is the base pairs themselves. Again, looking at this Venn diagram, A, G, and C, adenine, guanine, cytosine, they have all those in common. No big whoop. RNA, however, has a brand new nitrogen base called uracil. DNA has one called thymine. So again, that is one of the critical differences between DNA and RNA. uracil versus thymine. Also, we said that there was single strand versus double strand. And then we said that deoxyribose sugar versus a ribose sugar. Those are our three critical differences. So that brings us to what the heck is RNA even for? We said that we needed it for transcription and translation, but what does that mean? Okay, what is the function of RNA? Well, the function is to make those proteins because, again, those proteins are going to do everything in our bodies that keep us alive, that bring us our traits. But proteins are made in the ribosomes. If you recall from when we did cellular structure, we said that ribosomes were little protein factories. So proteins are actually made in the ribosomes. The problem is the ribosomes are in the cytoplasm. The DNA tells what proteins need to be made. That's great, except for the DNA is in the nucleus. Nucleus, cytoplasm. They don't really mix that much. That is a problem. So how do we solve this problem? Because the DNA is not leaving the nucleus and the ribosome is not going into the nucleus. But they have to meet somehow. How we solve this problem is through RNA. RNA, remember, it's smaller. It's a single strand. And it's not even the whole genetic code. It's just copying little pieces at a time. It's only copying a gene at a time. It's not copying the whole DNA code. That makes it smaller. Because it's so much smaller, that RNA molecule is actually capable. It's small enough to slip out of the nucleus. So it goes into the nucleus, is able to read the DNA, and then it can carry that message out to the ribosomes. All right, so a gene is copied to the DNA molecule. The gene on the DNA molecule, I should say, is copied. The copy of that DNA is what we call RNA. All right. The copy of those instructions are then sent to the ribosomes out in the cytoplasm. They're able to escape. They leave the nucleus and go to the cytoplasm to hang out with the ribosomes. RNA carries this message from the DNA, again, which is in the nucleus, to the ribosomes, again, in the cytoplasm. And now, because that message got taken from location A to location B, we are now able to make the proteins. The ribosome now knows which proteins to make, and it knows how to make them. Super critical. In order to carry out this process, we are going to need three types of RNA. Yes, there are three types of RNA. Number one is messenger RNA, or mRNA. Not super original, but it's what it's called. Messenger RNA is literally going to travel from the nucleus, where the DNA is, out into the cytoplasm, where the ribosomes are, with instructions. It's a messenger. It is like literally a messenger. Messenger RNA is the messenger. It is the go-between the DNA and the ribosomes in the cytoplasm. So whatever gene we're trying to make, it reads the DNA, it makes a little copy, and then it takes that copy and it tells the ribosomes, hey, buddy, this is what y'all need to do. There is the second type of, actually, let me back up 1 second. I do want to point out something. You ever notice how every time I write up DNA, I always do it in chunks of 3? Like I'll be like AAA, TTT, CCC, GGG. I always do it in groups of 3. There's a reason for that. A group of 3 nucleotides is something we call a codon, and that is going to become very important next week when we start talking about protein synthesis. So I just figured I'd point that out because I don't think it's officially here in the notes anywhere. All right, our second type of RNA is tRNA, or transfer RNA. Transfer RNA has an anticodon. In other words, it's like complementary to the codon. So if my mRNA says AAA, then my tRNA would say UUU. It is the anticodon. It's the complementary codons. All right, so each tRNA has an anticodon, which you can see at the top there, like the GCA in the picture, and then it has a binding site, okay? The transfer RNA reads the message on the mRNA, and then it goes and finds the amino acids to make the protein. Remember, remember how like nucleotides are the building blocks for nucleic acids? Amino acids are the building blocks for proteins. So it is tRNA's job to read the message and go, oh, okay, I'm going to go gather some ingredients. And it's going to go and gather the correct amino acids. And it is then going to transfer those amino acids from the cytoplasm to the ribosome. It is literally going to go out and get the ingredients and bring them back to the ribosome so it can make those proteins. And it does these 3 at a time, or one codon, or if you want to think of it in terms of tRNA, an anticodon at a time. That codon becomes very important, like I said, next week, those groups of 3. And we'll talk about what that means later on. All right. Third type of RNA, not too surprising, is called ribosomal RNA, or big surprise, rRNA. Ribosomal RNA is found in the, again, shocker, ribosome. Ribosomal RNA basically are the factories, right? They are used to bind the mRNA and the tRNA to the ribosome, and this allows all of our components, right? We have the message, we have the ingredients, and it's going to let everything come together to build the protein. And again, here in the picture, you see the RNA bringing in these little circular amino acids, and the ribosomal RNA is actually linking all of these amino acids together according to the rules brought in, or the instructions brought in by the messenger RNA. So we can see the messenger RNA is sort of the thing that looks kind of like DNA. The ribosomal RNA in this picture is this blue blob, and the transfer RNA in this picture is the thing that kind of looks like a plunger with a dot on top. All right, so transcription. Remember, to make a protein, I have to transcribe and then, transcription and then translate this information from the DNA. So, process of transcription. Number 1, please be mindful that transcription is a process of forming a strand of RNA from a strand of DNA. This process happens in the nucleus. Remember, DNA can't leave the nucleus, so it has to happen in the nucleus. All right, so the cell must make RNA in order to send this information to the cytoplasm to tell the ribosomes how to make proteins and which proteins to make. Now, an RNA's molecule is basically a very good, amazing, faithful copy of the gene's proteins building instructions. This RNA that goes from the DNA out into the ribosome is called our messenger RNA. Transcription continuing. So there is something called RNA polymerase. An enzyme called RNA polymerase catalyzes this reaction. Now remember, when we talk about catalyzation, what we are talking about is it's going to speed up the reaction without becoming part of the reaction. The purpose of transcription is to copy one gene at a time, just one gene from the DNA molecule. It might be the gene for your brown eyes. It might be the genes for your freckles. It might be the genes that makes you tall, short, whatever. But it is that one gene that we are going to be building. So where does one gene start and the next gene begin? Well, we have a promoter, and that is on the DNA sequence where the RNA polymerase attaches and starts transcribing. And then we have a terminator. And the terminator is a sequence of DNA that says, yep, that's the end of the gene. So there is a promoter that says, hey, start your RNA here. And then there is a terminator that says, nope, this is where the gene ends, stop here. This is already coded gene by gene on your DNA. So, RNA polymerase basically binds onto a site of DNA where at the promoter. RNA then separates the DNA strands, just kind of pulls them apart for a little bit. It then reads and builds the RNA. The one strand of DNA that it's using is we call the template, and then we build new nucleotides are inserted according to the base pair rules. We know this already. Adenine, if we're making RNA, is going to pair with uracil. Cytosine is going to always pair with guanine. And you'll notice in the picture, there is an active strand and an inactive strand. The side that's not being used, I mean, you can attach RNA to either side, it wouldn't matter. But the side that's not being used is the inactive strand. Big surprise there. When it reaches the end of the gene, we have reached the terminator. When the RNA polymerase moves along the DNA molecule, hydrogen bonds between the two strands are again reformed. So it basically comes in, makes a copy, closes things up, and moves on its way. And now we have a single strand of RNA molecule that has been transcribed. The word to transcribe means to copy exactly. There are people called transcriptionists that work in courts. And if maybe you've been to court, maybe you have a family member that works in a court, maybe you've seen this on TV. Those people with their little tiny keyboards in the courtroom that type really super fast, they are making an exact copy of everything that's being said in the courtroom. When we say RNA has transcribed the DNA, what we mean is it has made an exact copy of the DNA. Because again, we need an exact copy of what proteins we need to make and how to make them. Now remember, again, the purpose of transcription is not to copy the entire length of DNA. That would take forever. The purpose of transcription is to just copy a single gene. All right, so we are just copying one single gene when we go through transcription. mRNA transcript is now going to be sent out into the ribosome with the instructions for protein synthesis, with the instructions to how to build a protein and what protein to build. So, the RNA, however, is still not ready to be sent out in the cytoplasm because it has to be modified before it can serve its purpose. We talked about this in our interactive. The mRNA it's just a copy of a very small section of DNA, and is going to contain sections called introns and other sections called exons. Okay? The thing is, the introns are sequences of nitrogen bases that are not involved in making the protein. They're not necessary. So they basically need to be cut out of the RNA before the RNA goes to the ribosomes. The exons are the sequences of nitrogen bases that are involved in making the protein and therefore are very much necessary, and therefore those are the sections that have to go out into the ribosome. So again, up here we see the mRNA is formed. Both introns and exons are copied. However, the introns are cut out and of the RNA, and it still will contain the exons. So again, we can see up here the exons are in blue, the introns are in yellow. It will copy everything, but the thing that goes out to the ribosome is just going to be this blue code of just exons, because that is the necessary piece. Those exons are then spliced back together. You put them together, chunk them up, and shazam-bazam, we have our final RNA. We have the final RNA that now is going to go out in the cytoplasm because the DNA can't leave because it's way too big. It's all big. RNA is going to sneak out and it is going to hang out with the ribosome and make your protein so you can have those big, beautiful brown eyes of yours. All right, so that is the end of the notes. If you guys have any issues or questions, make sure that you ask. Other than that, we are really close to the end of class, so I just need to double check attendance and check in with a couple of you guys.